Okay, so welcome to this next video in which we are discussing the drug treatment of rheumatoid arthritis. Okay, so we're in the process of discussing the glucocorticoid drugs and how they're going to work. So we've seen that glucocorticoids come into cells and they bind to glucocorticoid receptors which are in the cytoplasm of cells. They cause the dissociation of the glucocorticoid receptor from the other proteins that it is usually in a complex with, and then what happens is that these glucocorticoid receptors which have the glucocorticoid bound to them then dimerize to form glucocorticoid receptor dimers. These glucocorticoid receptor dimers then go into the nucleus and are going to affect the expression of proteins. Basically, they're going to affect the expression of genes, and we've seen that glucocorticoid receptor dimers can act as transcription factors, and they can act as transcription factor enhancers and also as transcription factor repressors at different genes, basically. So, we've therefore seen a mechanism by which glucocorticoid receptor dimers can increase the expression of a gene, and we've also seen a mechanism by which they can decrease the expression of the gene by acting as a transcriptional factor repressor, basically. Okay, now we're in the process of looking at another way in which glucocorticoid receptor dimers can increase the expression of certain genes, basically. And they can increase the expression of genes by loosening the DNA, which is wrapped around histone octamers. Okay, so DNA is not just free within the nucleus of cells, instead it is wrapped around these histone octamers that we've described here, and that structure of DNA wrapped around a histone octamer is known as a nucleosome. Okay, now if the DNA is wrapped around the nucleosome very tightly, then basically RNA polymerase cannot get in to bind to the promoter regions of the genes that are within this portion of DNA which is wrapped around this histone octamer. Okay, therefore the genes that are on this stretch of DNA which is wrapped around this histone octamer will not be being transcribed, and therefore you will not be getting any protein from them. Okay, so another way that glucocorticoid receptor dimers can increase the expression of certain genes is by loosening the DNA that is wrapped around these histone octamers so that it becomes looser, and then the RNA polymerase II can then gain access to the promoter regions and transcribe those genes and then produce mRNA, and the mRNA can then be translated. Okay, so by loosening the DNA around these histone octamers, you can increase the expression of the genes that are within this stretch of DNA that's wrapped around the histone octamer. Okay, right, so how do you loosen the DNA that is wrapped around a histone octamer? Well, basically, if you acetylate the histone proteins, this leads to the DNA loosening around the histone octamer, basically. So, what we're going to do is construct histone acetyl transferases on the surface of the DNA, which will add acetyl groups onto certain amino acid residues, specifically on histone 4 in this case. So, what's going to happen is that the glucocorticoid receptors are going to bind to certain regions on the DNA that is wrapped around this histone octamer. Okay, so let's say this is a piece of the DNA that is wrapped around this histone octamer. What is then going to happen is that the glucocorticoid receptor dimer is going to bind to a portion of this DNA. Okay, so here are these two glucocorticoid receptors which are bound together. Okay, and I'll also show the glucocorticoids bound to them, but I'll put them on the sides now rather than on their tops because I want to put something else here now. Now, what is going to happen is another protein is going to come and bind to this glucocorticoid receptor dimer once it has bound to the DNA. Okay, and this protein is known as SRC1, and SRC1 stands for steroid receptor coactivator 1. Okay, so this is steroid receptor coactivator 1. So the S is for steroid, the R is for receptor, the C is for coactivator, and then the 1 is just for 1. So this is the steroid receptor coactivator 1. Okay, now 2 more proteins are going to bind on top of the steroid receptor coactivator 1 protein. Okay, and these 2 next proteins, one is known as the Krebs binding protein, and for short, Krebs binding protein is CBP. So CBP stands for Krebs binding protein. Okay, so BP is for binding protein. Okay, and the other protein which binds on top of the steroid receptor coactivator 1 is a protein known as PCAF, PCAF, and this stands for P300, okay, which we'll see in a moment, and then / CBP for Krebs binding protein. So the P is for P300 and the C is for Krebs binding protein, and then its associated factor. AF is for associated factor. So PCAF is the P300 Krebs binding protein associated factor. And then on top of the Krebs binding protein and the P300 Krebs binding protein associated factor, P300 then binds. Okay, so these 4 proteins, steroid receptor coactivator 1, Krebs binding protein, and P300 Krebs binding protein associated factor, and then finally P300, these all bind on top of the glucocorticoid receptor dimer. And this complex here, this functions as what is known as a HAT, okay? And this stands for histone acetyl transferase. Okay, so it's a histone, that's the H, acetyl, that's the A, and then the T is for transferase. Okay, so what is a histone acetyl transferase? Well, basically it is an enzyme which is going to add acetyl groups onto histone proteins, and this specific histone acetyl transferase is going to specifically add acetyl groups onto lysine residues in histone 4. Okay, so remember when we looked at the histone optimer, 2 of the histones in the histone optimer were histone 4 proteins. Okay, so onto histone 4, we are going to stick acetyl groups onto lysine at position 5 and lysine at position 16. These are going to be our targets. Okay, now let me just discuss the structure of lysine, and by the way, K is the single letter amino acid code for lysine, which is why I have put K5 and K16. So let me just discuss the structure of a lysine amino acid. Okay, so let's firstly start off with the core structure for the amino acid. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, and down here is the carboxylic acid group. Okay, now the R group for lysine, let me just straighten this up and bring it up a little bit. The R group for lysine consists of 4 methylene groups, and I'll show these. Okay, so all of these are methylene groups, which means that the carbons all have 2 hydrogens coming off. Okay, and then the final group on the end of these 4 methylene groups is then an amino group. Okay, so here is our amino group. Now, basically, if you want to transfer an acetyl group onto these lysine amino acids, which are within the within the histone 4 protein, then what you need to do is take an acetyl group, and this is the structure of an acetyl group. You have a carbonyl group there with a methyl group coming off, so this is acetyl. Okay, and this carbon obviously has one more bond that it needs to form. And of course, we get acetyl really from acetic acid. So here is acetic acid in full, what would now be called ethanoic acid rather than acetic acid. And what you do is you take the the hydrogen off the amino group and the alcohol group off the acetic acid. You bind those together to make water, and then you bind the nitrogen to the carbon. Now, it's likely that this histone acetyl transferase doesn't actually work with acetic acid. Instead, it's probably more likely that it uses something like acetyl coenzyme A to get the acetyl group from. But this describes the basic principle that you're going to add this acetyl group onto the amino group of lysines on histone 4. Okay, and the two lysines that you're going to use are lysine 5 and lysine 16 on histone 4. And when you add these acetyl groups onto those lysine residues of histone 4, what will happen is the DNA will loosen. Basically, it will become less tightly wrapped around the histone octamer, and that then will allow RNA polymerase 2, to gain access to the promoter regions for the genes, okay, of this stretch of DNA that's wrapped around the histone octamer, and therefore you will start actually expressing those genes, whereas before, when you had it very tightly wrapped around the histone octamer, you weren't expressing them. So this is another way in which glucocorticoid receptors can increase the expression of certain genes. Okay, so we've now looked at the pathway which is going to occur within cells, okay, and we've understood that when glucocorticoids go into cells, they're going to change the expression of genes, and they can either increase the expression of genes or they can decrease the expression of genes. Okay, so we're now going to look at specific examples of cells that the glucocorticoids can go into and act on, and which proteins they're going to upregulate and downregulate within that cell type, and how that's going to help prevent rheumatoid arthritis. Okay, so let's start with the endothelial cells of the blood vessels in the synovium. Okay, so the endothelial cells of the capillaries, the endothelial cells of the terminal arterioles, and the endothelial cells of the postcapillary venules. These are extremely important because these are in charge of the inflammatory response. Fundamentally, the inflammatory response is about bringing troops out of the blood and into the interstitial fluid of the synovial membrane. Okay, now it is endothelial cells which control the movement of things out of the blood and into the interstitial fluid. So fundamentally, it's the endothelial cells that control the inflammatory response, and the change occurs on the level of the endothelial cells. Okay, so we've seen that in order for endothelial cells to undergo activation, i.e. to start producing molecules which produce vasodilatation, to start retracting their outer edges to create holes in the endothelium so that fluid can move out of the blood into the interstitial fluid and form an inflammatory exudate, and also the changes that require them, that allow them to recruit leukocytes from the blood, grab hold of leukocytes and move them across the endothelium. These require pro-inflammatory cytokines which are released, or pro-inflammatory mediators, because you wouldn't count histamine as a cytokine, but pro-inflammatory mediators that are released by sentinel cells. And we've seen three main examples. We've seen interleukin-1, tumor necrosis factor alpha, and we've also seen histamine. We also saw bradykinin and calladin as well, and we saw that bradykinin and calladin work in a similar way to histamine, i.e. they produce type 1 activation. Okay, right. So these cause type 1 activation of endothelial cells. Okay, and these two, interleukin-1 and TNF-alpha, those produce type 2 activation. Okay, now we've seen, I've discussed that type 1 activation is really the weaker of the two. Type 2 activation is the strong one. This is the one that makes, it improves type 1 activation hugely, and it really results in the inflammatory response. Okay, so one of the things that glucocorticoids are going to do in endothelial cells is that they are going to stop type 2 activation completely, or at least hugely reduce it. Now, how are they going to do this? Well, basically, they increase the expression of, so when glucocorticoids go into the endothelial cells, one of the molecules they're going to increase the expression of is a molecule known as the inhibitor of NF-kappa B. So I-kappa B stands for inhibitor of nuclear factor kappa B, which is often abbreviated to NF kappa B. Okay, so I'll just write out what NF kappa B in full means. It means nuclear factor kappa B. Okay, right. So let me explain why this is going to have any effect at all. Basically, interleukin-1 and TNF-alpha both cause type 2 activation within endothelial cells by activating the nuclear factor kappa B. Now, how do they activate it? Well, usually inside the cytoplasm of the endothelial cell, you would have NF kappa B bound to another protein. So here is the NF kappa B, the nuclear factor kappa B, okay, which is usually made up of two proteins. In fact, it's usually made up of P50 and P65, but we won't worry about that. It doesn't matter for our purposes. Okay, and usually this nuclear factor kappa B is bound to the inhibitor of NF kappa B. Okay, and the inhibitor of NF kappa B keeps it in the cytoplasm and stops it from going into the nucleus. Now, if NF kappa B was to go into the nucleus, it's a transcription factor. It will alter the expression of a whole bunch of genes, and it will create a whole bunch of genes that cause type 2 activation. So, for instance, COX-2 is an example of one of the genes it's going to upregulate. But there are others. The genes that are involved in retracting the edges of the cell, those are going to be upregulated by NF kappa B. Okay, the genes that are involved in creating the proteins that are needed to recruit many different leukocytes, they're going to be upregulated by NF kappa B. So there are many different proteins that will be upregulated by NF kappa B. Okay, so usually what stops this is the inhibitor of NF kappa B. So usually what has to happen in order for interleukin-1 and TNF-alpha to activate the NF kappa B, which will then go into the nucleus and cause this upregulation of the expression of these proteins that are going to cause type 2 activation of the endothelial cell and therefore cause the inflammatory response. What they have to do is break this inhibitor of NF kappa B off from the NF kappa B, basically. Now, what both of these lead to, they lead to obviously large signaling cascades, but the overall results of these signaling cascades are that you add a phosphate group onto the inhibitor of NF kappa B here. So I'll colour this in purple. Now, you might think, great, now what's going to happen is that phosphate group is going to cause the inhibitor of NF kappa B to break off the NF kappa B, but it's not quite that simple. When you phosphorylate the inhibitor of NF kappa B, it does not break off the NF kappa B, not instantly. Instead, what now happens is once you've added the phosphate group on, the inhibitor of NF kappa B gets another group added onto it, and this other group is more scary. Okay, so it now gets a ubiquitin group stuck onto it. Now, ubiquitin is not a group that proteins want to get stuck on themselves. Basically, things which get ubiquitinated get targeted for destruction by the proteasome. Okay, so a proteasome is quite a formidable structure in the world of molecular biology. Okay, it's a tube basically, and proteins go in one end and fragments of proteins come out the other end. It basically chops proteins into pieces. I'm always reminded of that film Chicken Run by Disney, I think, when I think of the proteasome. It reminds me of the chicken pie machine, where Mrs. Tweedy tells her husband that chickens go in one end and pies come out the other end. It's kind of like that. That's what it always reminds me of. But anyway, this is the proteasome. So basically what's going to happen is proteins which have ubiquitin stuck onto them, they bind to the entrance, okay, into the proteasome, and they get pulled in. So what's going to happen is this inhibitor of NF kappa B is going to come over here, and it's still got the NF kappa B bound to it. Okay, so here's the inhibitor of kappa B, and underneath is the N nuclear factor kappa B itself. Okay, and now what's going to happen is this protein is gradually going to be unwound. It's going to have its tertiary structure broken into pieces, and it's going to be pulled through this proteasome, and then in the proteasome it'll be chopped up into pieces, basically. Okay, and eventually you will pull so much of the inhibitor of NF-kappa B in that it will lose its binding site to the NF-kappa B, and then what will happen is the NF-kappa B will be free, basically. It'll be liberated and it will go off, so it doesn't get pulled through as well. Okay, right, so NF-kappa B is then free in the cytoplasm, and it will go off to the nucleus and activate type 2 activation. Okay, now what these glucocorticoids are going to do is they're going to hugely increase your expression of the inhibitor of NF-kappa B, so that you have a huge amount of the inhibitor of NF-kappa B just sitting within the cytoplasm, basically. Okay, now the instant that you activate these pathways via interleukin-1 and TNF-alpha acting on their receptors on the surface of the endothelial cells, and you phosphorylate some of the inhibitor of NF-kappa B here, and it then gets ubiquitinated and dragged through the proteasome, and that you liberate the NF-kappa B, the instant the NF-kappa B will get liberated, another inhibitor of NF-kappa B will come along and bind to that NF-kappa B. Okay, so basically your efforts were futile. You're not going to induce type 2 activation in the endothelial cell because you've got just too much inhibitor of NF-kappa B. And that's one of the things that these glucocorticoids are going to do within endothelial cells. They're going to increase the expression of the inhibitor of NF-kappa B and thereby block the induction of type 2 activation. Okay, so that's one of the major things that they're going to do within endothelial cells, and that's one of the major ways you're going to block inflammation, by stopping type 2 activation. You're also going to attack the actual outcomes of NF-kappa B getting through to the nucleus. So remember, we discussed that NF-kappa B is a transcription factor, and if it does get through to the nucleus, it's going to cause an increase in the transcription of certain genes. Now, one of the genes it increased the transcription of was the gene for cyclooxygenase 2, okay, which remember was this bigger, better cyclooxygenase, which was much better at converting arachidonic acid to prostaglandin H2 than cyclooxygenase 1. The prostaglandin H2 could then be further converted to prostaglandin I2, which then was secreted from the endothelial cell to cause relaxation of the vascular smooth muscle cells and therefore vasodilatation. Now, basically, glucocorticoids are going to decrease the expression of COX-2. Okay, so you're going to reduce how much COX-2 you're making. So, if producing more inhibitor of NF-kappa B and stopping the whole type 2 activation process from occurring in the first place wasn't good enough, you're also actually going to stop the expression of COX-2 as well. Okay, so it's going to increase the expression of inhibitor of NF-kappa B, that's going to go up. It's going to decrease the expression of COX-2. Now, there's another thing that you're going to do within endothelial cells, and this is going to attack type 1 activation. Okay, and basically what you're going to do is increase your expression of a protein known as annexin A1. Okay, now annexin A1 also has another name. It's also called lipocortin 1. Okay, now how does producing this new protein, producing annexin A1 or lipocortin 1, how does that inhibit a component of type 1 activation? Well, let me just get my drawings from a while ago. When we discussed cyclooxygenase inhibitors, we discussed that in type 1 activated endothelial cells, what happens is calcium goes up. Okay, this activates the cellular phospholipase A2, which then breaks down phosphatidylcholine into lysophosphatidylcholine and arachidonic acid. And arachidonic acid can then be eventually converted into prostacyclin, which will cause vasodilatation, which is a major component of the inflammatory response. Okay, right. Annexin A1 inhibits this enzyme cellular phospholipase A2. Okay, so when you increase the expression of this, you're going to block the cellular phospholipase A2. So you're not going to get the breakdown of phosphatidylcholine into lysophosphatidylcholine and arachidonic acid. So you're going to stop type 1 activated endothelial cells from producing prostaglandin I2, basically. Okay, so these two block type 2 activation, and we've also seen annexin A1/lipocortin 1, which blocks type 1 activation, or at least a component of type 1 activation. It doesn't fully block type 1 activation because there are other components of type 1 activation, but it blocks at least one of these components of type 1 activation, which is the production of prostacyclin. Okay, right. Now, we've seen the way in which these drugs are going to reduce inflammation, and if you reduce inflammation, then you're going to hopefully reduce the number of leukocytes that are being recruited into the synovium, and that might reduce your activation of the adaptive immune response, and therefore hopefully lead to a reduction in the propagation of this pathological process. Now, glucocorticoids are also going to act as immunosuppressants, basically. So they're also going to act by stopping the adaptive immune responses. So remember, we discussed that rheumatoid arthritis is this vicious cycle where you get inflammation, which causes adaptive immune responses, which then leads to more inflammation, which then leads to more adaptive immune responses, and it just keeps propagating forward, and you get more and more destruction of the joint and destruction of the bones that are involved in that, and it just gets worse and worse, basically. Okay, well, we've now seen a way of reducing the inflammation. Now we're going to see a way of reducing the adaptive immune response. So, basically, for this, the glucocorticoids need to act on lymphocytes, okay? So not only do the glucocorticoids go into the endothelial cells and act on the endothelial cells, but they also go into lymphocytes and act on lymphocytes. Okay, so, basically, firstly, we're going to discuss how they are going to block the activation of more adaptive immune responses. So let me get my pictures. Okay, so here's what we drew when we discussed how we were going to activate the adaptive immune response. Basically, glucocorticoids will go into CD4 positive naive T cells, okay, and they will decrease the expression of interleukin 2. So remember, in order for CD4 positive naive T cells to activate, you had to get signal 1 and signal 2, which came from the T cell receptor in CD4 gave signal 1, and the receptors for the co-stimulatory molecules, CD28 and CD40 ligand, gave signal 2. And these two together led to the production of interleukin 2 and the interleukin 2 receptor alpha. Now basically, if you've got glucocorticoids in the nucleus of the CD4 positive naive T cell, they are going to stop the production of interleukin 2, basically. And if you don't produce that interleukin 2, you can't then get signal 3, which activates the T cells. Okay, so that's one of the ways that glucocorticoids are going to cause an immunosuppressive action and stop the immune response. Okay, they're going to stop the activation of T cells, okay, and that's going to lead to immunosuppression. Okay, so immunosuppression. So they'll go into T cells and stop them from being activated, and this works not only for naive T cells, but also for memory T cells, which also require interleukin 2 to activate them. So, interleukin 2 is going to be reduced down in expression. Okay, and glucocorticoids also cause something that isn't very well understood, which is called glucocorticoid-induced apoptosis. And it causes this within lymphocytes, basically. Okay, so glucocorticoids go into B lymphocytes and T lymphocytes, okay, including plasma cells, which are making the antibody, remember, and basically they cause glucocorticoid-induced apoptosis. Now, what is this? Well, basically the T T and B lymphocytes start committing suicide. They start committing apoptosis. They die. Okay, and the mechanism for this isn't well understood, but it's likely that the glucocorticoids will increase the expression of some apoptotic molecule, potentially some molecule in the intrinsic apoptosis pathway that will activate the intrinsic apoptosis pathway, and then the B or T lymphocyte will commit apoptosis and therefore die. Okay, and in this way you can also reduce the production of antibody, and of course it's the antibody that is propagating the rheumatoid arthritis forward, so blocking the production of the antibody is going to help prevent that. We also should note that in order to get the destruction of the bone, you require these T helper 17 cells to be produced to get the cytokines that are being released by the T helper 17 cells. Okay, and if you're blocking the activation of T cells and also causing them to commit apoptosis, then that's also going to help to reduce the degradation of the bones that are involved in the synovial joint. Okay, so that's all for this video. In the next video, we'll turn our attention to other immunosuppressant drugs.